Hello and welcome to my presentation. For my Keystone, I completed the BA/MA track in biotechnology and biochemistry and molecular biology, and so this is a component of my GRS Symposium slides. Now, when I say the words agriculture, I imagine we all conjure a vision of something like this. This is a fairly generic cornfield. It's a stock image. And agriculture really is a crucial component of not only the US economy, but also the global economy. It accounts for nearly 6% of US GDP, and it also accounts for nearly 12% of an estimated American household budget. But what happens when this picture, the one we see today, moves to this picture as a result of climate change? Now, we all are fairly well aware of the effects of climate change and also the various effects that will lead us from a kind of green crop world to a brown crop world. But the two specific stressors that I've chosen to focus on are light and temperature, really the increased light intensity and the increased temperature and the effects that that has on plants. Now, we as humans have our own job in making sure that we are mitigating the effects of climate change, but plants also have a way of mitigating the stressors of climate change that affect them, and that's called a defense pathway. A defense pathway, as defined by the Environmental Molecular Science Laboratory, is any mechanism or metabolic process in place to help fend off and adapt to stressors. Now, there are a variety of defense pathways, and some of the more obvious ones that we're all well aware of is the concept of thorns or poison berries or even sticky leaves to prevent pests from landing on them. And these are all mechanisms to prevent and fend off predators, but they are also opportunities to fend off environmental stressors. And one of the plants that I was looking at has a very interesting way of doing this. For my project, I looked at Arabidopsis thaliana, which is a member of the mustard and cabbage family. I promise I will only refer to it as Arabidopsis from now on. And the reason I used it was A, because it's easy to grow, it grows very quickly, and has a small sequenced genome of about 26,000 genes, which I know sounds like a large number, but it is a very small number relative to genetics. And this crucial part of it being sequenced is important to the concept of being able to look at the wild type genes and all the wild type plants and also look at the mutant plants and see exactly what changed and how that affects how the plant's defense mechanism works. And the defense response pathway that I'll be looking at for Arabidopsis is the production of a compound called an indole glucosinolate, or an IG. Now, that doesn't really mean a bunch to you, but if you guys have ever had mustard greens or broccoli, IGs are responsible for that bitter taste. Now, there are kind of three main ways, and split into qualitative and quantitative ways, that we can study these activation defense mechanisms and, crucially, the production of IGs. It would not be a science presentation if I didn't force you guys to look at a diagram of a compound. But the first thing I'll talk about is my primary quantitative method of looking at it, which is through high-performance liquid chromatography, and it specifically focuses on finding an ideal concentration, or the concentration of IGs in each of our plant samples, and it uses the specific ring-like structure in IGs. Now, this ring-like structure has the capability to fluoresce when put under UV light, which means that we can pass it through a filter and then a series of pumps and analyze the fluorescence of the protein to determine an exact concentration, and that is kind of the basic premise of HPLC, or high-performance liquid chromatography. But there are also times when you need not necessarily a quantitative, long, and tedious method, but you can get by with looking at a qualitative analysis of these defense pathways. First thing I will look at, though, is our quantitative research. So you can see this is a bar graph, and I'll slowly open up the plants. But we have our Columbia, or our wild type. This is the Arabidopsis that you would see in the wild. And you can see that when grown at 21 degrees Celsius, which is the optimal growth temperature for Arabidopsis, it grows and has a certain concentration of indole glucosinolates. But then when you move it to 29 degrees Celsius, stimulating that environmental stressor of being moved to an increased temperature, you see a statistically significant increase in IG production. Now, we also looked at two different genes, specifically analyzing if they were the ones in control of that IG production pathway, or in any way played a role. And you can see that they do have varying effects that are talked about in my longer talk, but I won't be covering here because there's a long explanation. But you can also see that when both of them are removed, which is this bar all the way to your right, and the MIB34 and MIB51 double knockout, which means that both of those genes are not present in the genome, that indole glucosinolates are not produced by the plant. Now, getting back to that concept of a qualitative mechanism, this is the concept of beta-glucuronidase or GUS staining. And the way this works is through a reporter gene mechanism. I don't know if anybody's ever seen those mice that have been genetically engineered to glow, but this is kind of using that same concept of using proteins that confer one specific report or one specific kind of noticeable change, and adding it to a genome in order to get a certain sense. And so what we've done is use GUS staining, which follows this gradient where low GUS is a very translucent color or white color, and then high GUS is a very dark blue color. And we've built it so that there's a correlation where darker color means more endoglucosinolates are produced, and more translucent means that less endoglucosinolates are produced. So this means that in those plants that have been shifted to the 29 degrees Celsius, we would have been expecting to see a darker color, as we had just proven in our previous slide. And thankfully, that is exactly what we saw. In the 21 degrees Celsius, we see some amount of endoglucosinolates, just like we saw in the bar graph earlier. But when shifted to 29 degrees Celsius, not only do we see more expression, but we also see significantly higher expression in those darkened plants. One of the things we also noticed as a result of the way we did things in the lab was that we were shifting the color of light that the plants were being grown in. And so we wanted to make sure and eliminate the variable of light intensity. And so we looked at the plants when grown under a white light and then under a yellow filter that the white light was shining through. And you can see, just comparing the two rows, that those plants look about the same. The top 21 degrees Celsius that was grown under white light looks about the same expression as yellow, and vice versa. And we've also been in the process of doing HPLC to further confirm this quantitatively. But looking at conclusions, we can obviously see that Arabidopsis increases the defense compound, that Ig production, in response to a variety of those external and internal environmental stressors. And we can also begin to delineate which specific genes play a more significant role. So one of the things that we do look at in that bar graph is that MIB51 is induced by increased temperature. That is one of the genes. And MIB34 is also induced by increased temperature, but it plays a less significant role in the response to the environmental stressor. In contrast, just as we had hoped, because that is it was an error on our part that thankfully did not end up being too much of an error, the intensity of the lights plays absolutely no significant role in affecting how the plants grow and also the defense pathways, and it really is the genome and temperature of the plant that affects that. So that leads us to ask, why continue to study the defense pathway in plants? We know about Arabidopsis, why continue to study it? And there are kind of three main reasons. The first one is due to the plants being stationary or sessile. They can't run away. We as humans can run away from stressors, but plants cannot, and so they must produce these anti-herbivoric compounds, and it's crucial for us to know about them. Climate change also is exposing plants to new stressors that some of them we're aware of, aka the increased temperature and light I studied, but some of which we're not necessarily aware of. And that really is something that we want to continue to be able to model in our defense pathways. And then a final thing is, one of the benefits of working with plants and working with conserved evolutionary genetics is that this very same findings that we're finding in Arabidopsis can also be applied to a variety of other plants and those staple crops that we know so well, like corn and wheat, as I was showing you in the beginning of the talk. So that is what I have for you. I'd like to give a special shout out to my PI, Dr. John Salenza, and my master's student, now PhD student that I work so closely with and train me, and is the absolute best, Erica, who's in the back of the room, and then the other, my other peers in the lab. Any questions? Thank you.
Okay, any questions for Tyler?
Hi, Tyler. You did a great job presenting, and I was wondering how you can use this information to apply to specific agricultural techniques.
Yeah, so it's twofold. One, this is confirming mechanisms by which we can study defense pathways, and it also is a lot of the genes that we're seeing in that MIB51 and MIB34 expression in the entire pathway. While they might not be directly conserved, a lot of the mechanisms will remain the same across all plants, and so it really is what we can study here, which we can study more easily because we have the full genome sequence, and so we know what a wild type is supposed to look like and what changes when the defense pathway is activated can be applied to a variety of plants.
Any other questions?
Hi, I was curious to learn a little bit more about like why this plant specifically, and also kind of where it originates.
Yeah, so I'm not going to lie, I don't know exactly where Arabidopsis thaliana originates from, but the main focus is the fact that the vast majority of all living organisms, we don't have a completely sequenced genome of. And so any changes to that genome, we don't really have a mechanism of confirming. Whereas Arabidopsis, we have the wild type genome, which means that any deviations we can analyze and compare back and forth. So it just makes it a little bit easier to do that analysis and find exactly what's changing. Great question.
All right, time for one more. 2 more, Adam? How many? One more? Okay.
Hi, thank you for your presentation. I just had a question about the temperature change in general. So when you increase the temperature, you see that you have a higher induction of IGs, right? But have you tried growing them at a higher temperature and then lowering the temperature to see if that changes recapitulated or not?
So we haven't tried growing them at a higher temperature, or at least I haven't tried in the lab growing them at a higher temperature and moving them to a lower temperature. We have grown them at a constant high temperature, and we also see IG induction. One of the difficulties with the way that HPLC and gust staining work, at least though, is that the indoglycosilates will still be present in the sample, and so where that plays a role might skew our data, and so we wouldn't necessarily be able to see like a marked decrease based on a shift down. But that's definitely something to study further, and there are definitely mechanisms to do that test.